Hello everyone, we are from group 12. We're gonna explain and present about cultivating rabies virus with various types of culture media and virus purification technique. This group consists of Rahma Amalia Saraswati and me Satria Badani. The taxonomy will be explained by Rahma.
Okay, so first we're going to describe the classification of Rhabdoviridae. The subfamily is the Alpha Rhabdovirinae. The genus is Lisa virus, and the species of this virus is Rabies Lisa virus. The disease that this virus causes is rabies in humans and animals. There are a few methods to culture this virus. One of the culture method of rabies is called the rabies tissue culture infection test, or the RTCIT. The process includes the addition of a suspension of suspect brain tissue to a suspension of a murine neuroblastoma cell in a 9612 microliter plates. The culture were incubated at 35 until 36 degrees Celsius for 4 days, and when it's ready, it is stained with a fluorescein labeled hamster anti-rabies antibody conjugate and examined with a fluorescence microscope. The medium used is 10% brain suspension in a physiological saline containing 500 IU of penicillin and 2 milligrams of streptomycin sulfate per milli. If present, rabies virus will appear as bright apple green inclusion in the cytoplasm of the infected cell through the fluorescent microscope. Next, the method will be explained by Satria.
Okay, so the second method of viral culture method is a method that carrying out the DFAT test. So first, the virus samples were suspended at 10% with PBS containing 2% fetal bovine serum, and the virus was isolated in hamster kidney cells and neuroblastoma cells. The sample was inoculated 0.2 milliliter in the kidney cells, and the culture was then incubated at 33 Celsius degree for an hour. Then growth medium was added and incubated again. 4 days after inoculations, media was harvested and stored at -60 Celsius until use. Neuroblastoma cells that had been inoculated with rabies virus isolate were prepared on an optic glass and incubated at 37 Celsius degrees for 2 hours. Cells were then washed with PBS 3 times and fixed with acetone for 10 minutes at room temperature. After incubation, the slides were washed with PBS and added with 50 microliters of immunoglobulin G-GOT antinucleoprotein rabies conjugate and incubated for 30 minutes in an incubator at 37 Celsius degree. Next, the slides were washed with FA rinse buffer pH 9, soaked for 10 minutes and dried. Before the slides were dry, they were mounted with the addition of FA mounting fluid and read with a fluorescence microscope at several magnification. Next, please. The culture medium that is used in this method is hamster kidney cells, or PHK21, and neuroblastoma cells. And if the result is positive, the CPA occurs in the form of cell lysis. The emergence of CPE indicates that the rabies virus has adapted and is growing well. To prove the presence of rabies virus, identification was carried out by serological tests, namely indirect sandwich enzyme link immunosorbent assay and direct fluorescence antibody technique. Next, please. This is the picture of normal NS cells and infected NS cells. Next. So the life cycle of rabies virus, it will attach to the host cell wall. Then by endocytosis, the virus is inserted into the host cells. In the penetration stage, the virus enters and unifies itself with the host cells. Then transcription and translations occur. The structural compartment of the virus has been formed, followed by the process of assembling and releasing the virus from the host cells to infect other hosts. The whole process in the life cycle of the rabies virus takes place in the cytoplasm. Next. Okay, so that's all from us. Thank you.